Okay. I think I have this going. Um, yeah. Okay. This was a little hard, so I haven't gotten everything. Let's see here. Make sure I have the right one. Okay. Yeah, so I'm having to do this live today because let me scoot here where I a little bit more in my frame here. But at any rate, on the way into taking my kids, my daughter got really sick. She was throwing up on the way into school, so there's no way I can take her to daycare. So let's do this live. Much rather be in class. All right, so here's what we're going to talk about today. So what we're going to do is we're going to continue on about evolution by natural selection and evolution in general. We've covered natural selection a lot, but there's this one interesting part of natural selection called sexual selection. So let's take a look here. Let's see if this will work. We can see my slides. Yep. Okay. So mechanisms of evolution. We know that natural selection causes adaptive evolution. And there's also other mechanisms of evolution, including genetic drift, gene flow, and mutations. Now, interestingly, in natural selection, there's this thing called sexual selection. Now, sexual selection, it kind of causes adaptive evolution, but it really improves your fitness because it improves your ability to reproduce and pass on your genes to the next generation. Although some of those adaptations might not necessarily be there to help you survive in the environment, right? Which is interesting in itself. Okay, so as we go through, I see some of you are coming online now. You can always hit me up in the top chat if you have any questions. And let's get started with this sexual selection here. This is a really interesting thing. And for those of you that are interested, Kelly Miller, I think I mentioned this yesterday in class. He teaches a fantastic course on sexual selection, and of course Randy Thornhill every fall teaches a class on sex as well. All right, so sexual selection. This is a type of natural selection where one sex chooses specific traits of the other sex. So I've got a green darter, that's actually a fish in New Mexico. I've got those male turkeys strutting, and on the far right you can see that lizard. That's a Nolus cuniale, discovered by our own Steve Poe and some of his graduate students, Eric Kulabak, 15, 20 years ago in Panama, and then rediscovered Anolis proboscis. He's got the long nose on there, and basically these are all sexually selected traits that they have. So, and of course, obviously the peacocks. I mean, these are like the epitome of sexual selection. Okay, so, when we talk about evolution of sexual selection, there's a lot of papers about how it works, the importance of it, examples of it all over the place, from these dragonflies to these males, horseshoe crabs choosing that one female to that male and that female line. What's going on? Why are we all reproducing sexually here? And this is interesting here. Why reproduce sexually? And why have two sexes? Now, this is really important. And before we get into the nuts and bolts of how it may have evolved in the first place, let's do this. Let's make sure we know what sexual selection is. So sexual selection, we're going to combine two genomes to form a new individual. So, of course, there is an egg being fertilized by the sperm. The egg has 23 chromosomes from mom. Sperm has 23 chromosomes from dad. And then as a result, when you combine those two genomes into a new individual, you have a new organism. That is sexual reproduction. And you do this by reproducing with gametes. So of course, this leads to two sexes, male and female. And across the animal kingdom and with plants as well, fungus are weird. But basically, we've got two sexes that you can basically tell because the male has the much smaller male gamete, the sperm, which is basically just a genetic donor, and then the female, has a large egg that has that has all the cytoplasm and organelles, including the mitochondria. And of course, Willow is saying hello. Willow, let's go back in there and watch TV, okay? We're going to say hi to your mommy, okay? Aww. Hang on one second. This is what happens when we go live here. Come on. Okay. Yeah. Yeah. Okay. So let's get back to this. So, now that we know that we can almost always identify male and female based on the size of the gametes, especially with plants and animals, like I said, fungus are a bit weird. We know that we've got these two sexes that reproduce sexually. But the question is, why did they evolve in the first place? Like, why do this, right? I mean, why split in half the number of individuals you can reproduce with? So not only do you have to go find a mate, you have to find the right mating type, which is an interesting thing, right? Because if you're asexually reproducing, well, and times are good, just reproduce a clone of you. And if you're doing good in the environment, then guess what? Your clone is going to do well as well. But when you do sexual reproduction, you have to find a mate. And because we have male and female, you got to have the right mate. So why do this? And also, why have the female with a very large egg and the male with a very tiny sperm? What's going on here? All right, so here's where we think the origins of sexual reproduction come from. Is the mitochondria. The mitochondria did it, right? So go back, 2 billion years ago. 2 billion years ago, we've got, this evolution of eukaryotes. Basically, you've got this aerobically respiring bacteria finds its way into a methanogen, I think, which is a type of archaean. And they merged and they formed this chimera, so to speak. And what happens is that we get the evolution of eukaryotes. And this is the largest restructuring of cells in like 2 billion years of life. And we get these larger, more complex cells. And of course, this goes on to form all multicellular life. So these mitochondria are really good because what they're doing is they're supplying a lot more ATP to these cells. A lot. Okay? Because they use oxygen. It's much more efficient. So you get a lot more energy, and this is a good thing. Okay. But there's a trade-off to using oxygen. There's a big trade-off, right? One of those is you can produce what's called a reactive oxygen species, an ROS. Usually I like to make a joke about ROUS as in rodents of unusual size here, but you've got these reactive oxygen species. Now, these things will basically, what they'll do is they can damage proteins. They can damage your membranes. They can damage your DNA, right? So all of these things can cause damage to your cells, all of this oxygen. So, hey Willow, she's wanting to play here. So, with the mitochondria producing all this ATP, the cost is it's causing cellular damage by causing oxidation events. So, how do you get around that, right? What do you do? Okay, yeah. All right. So what do you do here? Well, if your DNA gets damaged and you damage the wrong gene, and you've only got one copy of that gene, and it's an essential gene, then what happens is that cell dies, and all the mitochondria die with it. So it's gone. So one way we can do this, okay, is let's imagine you've got 2 copies of your genome. So that way, if one critical gene gets damaged, right, if it gets damaged, you've got a backup. Not only do you have a backup gene, you've also got a gene that has a functional copy of which you can use to repair. It can be a template to repair the other gene, right? So, and if you remember, sexual reproduction, we defined it as a merger of 2 genomes to form a new organism. So having that 2 genomes is a backup, right? It's a backup in case DNA gets damaged by having lots of mitochondria. Now the question is, why do we have these 2 sexes? And the answer with 2 sexes is, all right, Willow. The answer is, okay, if I'm merging 2 cells and I merge all the mitochondria, then those mitochondria have different lineages, which means that they would begin to compete against each other. And if they're competing against each other, that's not good. But if they're all clones, then they're all genetically identical, then we're okay. All right? So, by having the evolution of female, you get all your mitochondria, so all going to be genetic clones, you get all of those from the female. That's perfect, right? And then the male, all he does is just basically donate genetic material, donating genes. So this is why we think that evolution of sexual selection occurred was in response to mitochondria. Not only did it lead to this combining of two genomes, it also led to the male and female, so that the mitochondria, you'll have all your mitochondria from one parent, so they're all genetically clones and they don't compete against each other. So that's the origins of sexual reproduction. And for those of you that are really interested in this, there's a book called Sex, Power, and Suicide, Mitochondria by Nick Lane. And here's the name of him right here. Nick Lane is one of my favorite authors, especially for the cell and molecular folks. But he did Power, Sex, Suicide, and the Mitochondria. Fantastic book that, really shaped the way I think about a lot of these things as well. So I'm going to post that right there in the top chat so you guys can have that. Like I said, it's a fantastic read. Okay, so now we have this idea of we have this pretty good working theory of why we've got two sexes. But now the next thing comes is well, why have sexual selection? Why is one mate choosing somebody for something else? And there's a lot of theories out there about why, and the answer might lie somewhere with a little bit of all of them. But one of them is energetics. One of the reasons why we think that males tend to be more ornamented than females is because, for one, it takes energy to be ornamented, right? But also the cost of making eggs, the cost of those female gametes is more than the cost of making sperm, right? And not only that, a lot of times females do parental care as well. Sometimes males do. There's some fish that, the female just leaves all the babies with the male. Okay, so if you look at this kiwi, look at the size of that egg, right? I mean, that's enormous. And that egg is 20% of the female's body weight. So that's an enormous cost of being a female and producing more expensive gametes. The other reason why we think that males tend to be more ornamented is that it shows the good genes. This is called the good genes theory. So, for example, if you're showy, look at that male wood duck. Those feathers are beautiful. They're healthy. That tells you a lot about its health. That wood duck is basically disease and parasite free. It's getting food. It's avoiding predation. So as a result, it's showing to the female, I have good genes. You want my genes for your offspring. Okay, there's other examples of that. Let's see here. Oh yeah. Okay, so I had to remember the pronghorn one. I couldn't remember the exact study here, but in 2006, what was interesting is that they discovered that the offspring of good-looking male pronghorn were more likely to survive past the weaning age and survive later into 5 years. So the good-looking males, whatever they were judging on that, probably symmetry, probably the shininess of the coat, the health of the horns, things like that, parasite loads, ones that had, the traits showing that they were good, they're healthier, they have better genes, which led to, better offspring. All right. Also, a lot of times in sexual selection, we see this ornamentation, and here's these flamingos. Hold on. Flamingos, right? They're bright pink. And basically, these flamingos are enhancing their color. And they're doing it by rubbing carotenoids that they get from their parietal gland onto their feathers, making them basically more brightly pink. Okay, now, Ronald Fisher, one of those members, or not members, but one of the important scientists of the modern synthesis. He was a great statistician. Well, one thing he postulated about sexual selection was that you could have runaway or Fisherian selection, a Fisherian runaway. And the idea is that the females keep selecting for these traits, and the males just get more and more and more ornamented. And of course, to me, the peacock is a prime example of it. I mean, the colors and all the different parts of them that are so colorful. And then their tails. Those tails are enormous. So eventually with runaway sexual selection, eventually you're going to get checked by natural selection. You're going to become so ornamented that you might not be able to forage for food. You might not be able to fight off diseases. You might get nailed by a predator. And when we look at the peacock's tail, if you look closely at the tail, you'll see that it looks like it has lots of eyes. So if you're a fox or some bird or some predator about to jump on a peacock and they expand that tail rapidly, not only does a peacock go from looking one size but much larger, but it looks like it's got a bunch of eyes looking at you as well, which could actually ward off predators. So not only is it helping with sexual selection, it could be helping with predation as well. Other examples of Fisherian runaway, and we know that the ladies, the female birds, like the males with the longer tails. And then they've done experiments where they've actually cut the tails off of some, added tails to other, and clearly the females choose the males with the longer tails. And the one on the second left, that is actually a scissor-tail flycatcher. It's got the kind of the green background. That's from North America. We have lots of those here. Okay. Oh, runaway selection, runaway Fisherian selection in fashion. You know, we laugh. I mean, these are the hair bands of the 80s from Poison and Cinderella and Motley Crue and Twisted Sister. They were looking a little on the absurd side with their hair and their makeup and all of their fancy clothes and things, you know. But this really shows that very rapidly in the 1980s that fashion quickly went one direction, almost in a Fisherian runaway selection. We look back at that and we're like, man, what were they thinking? Sounds like a country song to me. But, you know, that was the style back in my formative years, you know, so I still really like some of this music. However, this was also in my formative years, and the hair band ended on a dime, basically, when Nirvana released Smells Like Teen Spirit in around 1990s, or 91 or something like that. They did Smells Like Teen Spirit, and I mean, hair bands were gone, and that was it. I mean, they died overnight almost. So what this is also showing, we laugh at these fashion trends here in humans, but it also shows that even in nature, that female choice can start on, it could be almost anything they're going after or what they're choosing. It could be color of a fin, it could be color of a feather, it could be a doohickey on the top of the head. But those changes, those choices can also change quickly, much like our fashion. Okay, so let's take some examples of sexual selection. The one on the left, this is an anolis lizard. You can see the dewlap. That's a skin flap right there. They're used to ward off other males and also to attract females. You can see the proboscis on Anolis proboscis. And then on the right is our Scalapris again. I can't remember that's Clarki or Poinsettia or whatever they've decided those are down there now. And he's got the bright colored underbelly and dewlap as well. All of these colorful patterns are there to attract females. And in fact, the Anolis lizards, there's like 400 species or so of these guys. And all of them have dewlaps that they use to attract females. And the dewlap color is really species specific, which is interesting. So not only are they using their dewlap, but the coloration, the patterns of that is important for speciation in these things as well. Then, of course, we see a lot of sexual selection in birds. We often see brightly colored males, but not just in the feathers. On the left, you can see what is called a magnificent frigate bird. You can see this enlarged gular patch, which is bright red with black flecks in it. The red-capped manakin, they dance. If you ever watch a manakin bird, just go YouTube it when you get done with this. But the manakin birds are dancing, right? And they literally doing all these funky dances on the branches. And then a northern mockingbird. The males and females are practically indistinguishable from each other, but the males attract the females by their songs. Okay. We also see sexual selection in fish. On the left side, we got the green darter, and then going across the top, the red shiner. That's from the Rio Grande River. Oh, that was from Bitter Lake, sorry, but we also have them in the Rio Grande River and Inler's live bears. And you can see clearly that when you start seeing colorful fish, well, yeah, those colorful fish patterns, that's an example of sexual selection, especially if the male is different from the female. And then on the bottom are the cichlids. And we think that sexual selection has been really important in driving the evolution of cichlids. And there's a lot of different species of cichlids in both South America and in Africa, and they're actually like a really model of like evolution that we study and how cichlid evolution was so fast. Okay, but sexual selection in mammals as well. Well, we've got elk and deer, a lot of those artiodactylus, or cervids actually. They've got the large horns that they're used to fight with. Those are elk fighting from California. Lions, they have a mane, and that mane probably serves two purposes. One, the ladies like the guy with the big mane. But also, if lions are biting each other and fighting, then that really thick patch of hair can actually help protect them from fighting. So this is a lesser prairie chicken from New Mexico, and there's a lot of traits on this bird. It dances, it's singing, it's got the way it's holding its feathers, it's got these eye combs, it's got head feathers, it's got different types of tails. Now, this bird cannot go out and look like a peacock because it lives out in the grasslands. So here you've got this balance between sexual selection, where the females are clearly picking up on all these different traits. They're portraying good genes, but they can't get as wild as like a jungle bird because these birds have to live out in the open and they have to still blend in with the environment because they're very open to like birds of prey like hawks and owls. Okay, so let's take a look at some of the differences that we see in in our sexual selection. And I'm going to show you, this is a mockingbird song. I recorded this. I think it will work. Let me see if I can get this recording to work. I have it on YouTube. But I made this recording back a few years ago from St. George Island in North Florida. Let's see if this will come through. All right, that's the northern mockingbird calling for a female. He was actually quite cooperative and let me get right up to him there. Okay, next. Okay, sometimes females like to have a male that can sing, but also have nice feathers. And this is the lyre bird. And you guys, if you can't hear this, let me know, okay? And I'll see if I can adjust the volume. But this is from the BBC with David Attenborough here. Okay, so it looks like the sound might not be coming through here. I'm not sure how to fix that problem. Okay, which is unfortunate. And I'll show these in class when I can come, probably tomorrow when I come back. But this is a Western grebe, and I'll show this video, like I said, in class. And another one example of sexual selection. It doesn't have to be how bright you are, but it can also be the beautiful feathers as well. And this, of course, is the greater sage grouse. And this photo is taken by a friend of mine and former student, actually, Neil Lowson. He's one of the most fantastic photographers I have ever known. And that's one of his photos of a greater sage grouse. And I have a video of it, which, like I said, we'll show it in class later. Much of sexual selection focuses on female choice, where females are choosing traits for males. And surprisingly, throughout the animal kingdom, male choice is actually rare, except in humans. And in humans, male choice, well, parts of what we consider attractive on many females, and Jessica Rabbit here from Roger Rabbit shows the accentuated traits that is often selected for by men, okay, including hip to waist ratio and adipose tissue around the mammary glands. So these are examples of what would be male choice. And Randy Thornhill at UNM also teaches a class on human sexuality, and I think he's been teaching that class for about 20 years or so every fall. So if you get a chance, he's about to retire, I would take that class with him. And then funny on this, there's been scientific studies that show that yes, when women are around, men can actually become cognitively impaired. There's a really fun article in scientificamerican.com. I've posted the link in my slideshow, and it says why interacting with women leave men cognitively impaired. So, of course, I'm showing the late Betty White, who was quite spectacular in her. Okay. With sexual selection, we often get what is called sexual dimorphism. Now, di means two, morph means body shape, and in sexual dimorphism, this is where we often get, a larger female or a larger male. And in humans, males are typically larger than females, and my friends right here represent a very good example of sexual dimorphism in humans. My buddy was he was a linebacker. He played collegiate football as an outside and inside linebacker, and that's with his friend. And as you can see, he's quite clearly much larger than she is. It's an extreme case of sexual dimorphism in humans, but in general, men are larger than the females, and those size differences are due to natural selection. So in humans, it's been thought that we were hunter-gatherers, so men were larger, more muscular to do more hunting. That's just one theory. There could be more complicated than that. If you look at other animals, often the female is larger than the male. So in a lot of birds, the females tend to be larger than the males, especially in hawks, and that could be because of the energetics of child rearing. And it also, having slightly different sizes and sex allows you to go after slightly different prey. So you could actually do a little bit of intra-specific niche partitioning doing that. Okay. And then also male-male competition can lead to larger males. I cannot believe I got this photograph. This is in California at Point Reyes. It's literally in the sunset. These two male elk are fighting each other. And as a result of males competing for access to females because they're fighting, they tend to be larger than the females. Okay, so that's it for sexual selection right there. Now, let's go on to our next slide here. We're going to switch gears. Normally I would do a whole nother lecture video for this, but I don't want to start and stop again, so let's just keep rolling with this. Okay, before I get started, I'm going to just shut my door. I have my kids watching Frozen. I never thought I'd ever see that movie. Then I have kids, and now I see it every day. Okay, now we're going to move on to these other definitions, these other mechanisms of evolution. So we know that natural selection, including sexual selection, is the only mechanism of evolution that causes adaptive evolution, meaning you're getting more fit to the environment, or you're getting more fit to, specifically when we talk about fitness, that's how much offspring you produce. So sexual selection helps you attract the females, which leads to reproduction and more offspring, hence improved fitness. These other ones are the genetic drift, things like founder effect, bottlenecks, gene flow, and we're going to talk a little bit about metapopulations and different population structure, and then mutations, which of course is the ultimate source of genetic variation. So let's take a look here. Genetic drift, gene flow, mutation. These don't really cause adaptive evolution. They can, but that's not a driving force behind them. These are more or less random processes. So genetic drift. This is a change in the frequency of an allele in a population. It's due to chance. All right? So, hey, we're changing those allele frequencies. We know that that's a definition of evolution, right? But instead of it being for differential survival and increased fitness, this is just due to chance. And this was coined by Sewell Wright, another one of those figures of the modern synthesis. Okay, if you have an infinitely large population, remember that from Hardy Weinberg, then the allele frequencies will remain constant from generation to generation, unless the population is taken out of equilibrium by migration, mutations, selection, or anything like that. But no population is infinitely large. This means that it's possible for an allele to become fixed in that population, okay? And the smaller the population, the more likely an allele could be fixed. Now, let me back up just a tad bit here. In small populations, a common allele is more likely to become fixed. Now, what that means is, if you have an allele being fixed, so let's imagine we've got A, B, and O blood. Those are three different alleles for blood. Let's say we lose O and B, and we only have A. At that point, we would say that A has become fixed. So 100% of the population carries that allele. So if an allele is fixed, everybody has it. There's no other alleles, and you become a very homozygous, or you've increased homozygosity in your population. So homozygous means you've only got, one copy, one allele. So if we've increased homozygosity in a population, we're losing genetic variability because we're losing alleles. Okay? So, so smaller populations are more likely to have a loss of alleles. So, since we're losing an allele, believe it or not, that population structure is now different because you've lost your variation in it here. Okay, so all right. Okay, so the probability. Sorry, I'm hearing some squawking in the background there. Okay, so basically, like I said, the population, as it becomes smaller and smaller and smaller, you are more likely to lose genetic variability through genetic drift. So the important point here is that it's random with respect to fitness. These alleles could be fixed regardless if they're beneficial, if they're good for you, or if they're detrimental, or if they're neutral. So small populations this is most pronounced. Doesn't matter if they're good or bad, they can be fixed. Now, interestingly, and I usually ask this on a test, that genetic drift can be beneficial to a population if it randomly removes a deleterious allele. All right? So if you purge yourself of deleterious alleles, then you've actually increased the fitness of the individuals in the population, right? So that's important. But because this is a random process, genetic drift can also be bad because just as easily as randomly fixing good alleles and purging the bad ones, you could also fix the deleterious alleles, and then the individuals in your population start carrying a higher load or more of these deleterious alleles. So by fixing alleles, you also are reducing heterozygosity of your population. You're reducing genetic variability. And when you reduce genetic variability, you limit that population's ability to evolve, right? Because you need variation in the population. So by fixing alleles, you're losing variation in your population, and you hinder that population's ability to adapt to a changing environment, whether that's getting warmer, drier, hotter, colder, change in food source, change in predation, new diseases, any of that. And interestingly, an allele that might be bad under one circumstance might be beneficial under another circumstance. So if you change your weather and an allele that, is an adaptation for cooler environments, but it's lost, an adaptation for warmer environments and it's lost during a time when it's cool, it doesn't matter. But if you warm up, then that population can't respond. It can't adapt to it because you've lost the variability to allow you to survive in warmer environments. Okay, now next, genetic drift is I just saw your thing right there before we got there. Yeah, it's applied mostly to small populations. It can occur in a large population, but the larger the population, the less likely genetic drift will affect it. So I hope that I see you got it. All right. Okay. Now here's the thing with bottlenecks. So two ways here. We can have the bottleneck, which is you have a large population, and that large population gets small very quickly. And what happens is if you go from like 10,000 individuals down to 100 individuals, and it's quick, those 100 individuals might lack the genetic variability of your population that was just a few years back that was much larger. The other one is a founder effect. So the founder effect basically is if I've got a small flock of birds from the mainland and they fly and get lost and land on a new island, then that group of birds almost certainly has a different allele frequency, right? Because it's got a smaller, it's got some subset of the alleles from the large population and may not have all of them. So when we talk about bottlenecks, we cause this all the time in nature. Anytime we have populations that go from very large to very small in nature, we're causing this bottlenecking event where the new population has less genetic variability than the original population. So for example, you can see that you've got the white and blue and yellow, and you can see that the yellow is rare, but if I subsample a small portion of that population, then you're unlikely to get that yellow allele, and that's exactly that surviving population now doesn't even have that yellow allele. It's lost some of its genetic variability. So we see this in conservation of endangered species because your endangered species might have less genetic variability than the historical populations, which it hampers their ability to respond to the environment. So I'm showing prairie chickens. The greater prairie chicken has declined precipitously throughout much of its range, and the American bison, of course, declined from 60 million individuals in 1880, 1890. By 1890 it declined to like 750 individuals, but it was 60 million around, the early 1800s. So that is tiny. There's a lot more bison now, but the genetic variability of these populations has been greatly reduced because they've gone through bottlenecks. And then also, in addition to the bottleneck, you've got genetic drift through the founder effect, where you've got like a small population, like a flock of birds lands on an island. They have some subset. Now, in both bottlenecks and founder effects, because you're starting with small populations that have reduced heterozygosity, they have reduced genetic variability, genetic drift can act on these. It's more likely to act on these populations, which can cause problems for them. Okay. Now, genetic drift and the founder effect, these, of course, were developed by Ernst Mayr in 1942, and it's thought that the founder effect is important in speciation. And if you go around the world to all the different islands throughout the Pacific and whatnot, and the Atlantic too, and Caribbean, one thing you'll find, these islands have a lot of unique species, a lot of endemics. And it's thought, it was proposed by Ernst Mayr, that when these populations landed there, you're going through a bottleneck. There's genetic drift, but they can instantly start going down their own evolutionary trajectory and speciate rather quickly because they've got unique genes, or they will. Okay, the next one. So that's genetic drift. Is gene flow. Okay, gene flow is a movement of genetic variation. We're just going to move genes between populations. Now, interestingly, gene flow, by definition, can cause evolution because you're changing the allele frequency of that population, and the population is a unit of evolution. Okay, so here's an example of a mountain. You could have population A and population B, one on each side of those mountains. And basically, they're under different selective pressure, but a little bit of movement of birds between those two populations would represent gene flow, bringing in new variability. And like I said, it doesn't take much. Now, interestingly, although gene flow between populations is a mechanism of evolution, gene flow can also prevent evolution. And it can prevent populations from diverging. So as little as one individual a generation migrating between populations can keep those populations from diverging sufficiently to become unique species. So gene flow is interesting because, yes, it can cause evolution, but it can also keep your populations from diverging as well. Okay, so there's lots of factors that affect gene flow. Salamanders don't move around that far. Believe it or not, they're amphibians, but they don't have to always return to water to reproduce. Some of them can do direct development, so they live in these forests underneath the leaf litter and in wet logs, and they just lay an egg that directly develops into a little salamander. Because they don't move around much, you could just cut a road through a forest, and that's sufficient to half the populations, right? Where now you can start getting a reduction in gene flow in the population and almost separate them. In contrast, something like a pelican. Pelicans are far-ranging species on both the Atlantic and the Pacific. They fly very far, and they have high rates of gene flows between their different populations. So there's very little population structure there. Okay, and then of course, mobility and diversity, fragmented habitats, physical barriers. If you're a salamander, cutting down a forest, cutting a road can have a big impact. Also, distance. I've got the Pacific Ocean. It's enormous. And in fact, if I showed the Pacific Ocean from the South Pacific in the middle of it, you would think we're in a water world because you couldn't even see the continents. So the islands, it's dotted with islands way out there in the middle of it, that distance also can affect gene flow. So the further you are away from your populations, the less gene flow you're going to have. That's part of what we learned from island biogeography. Now, when it comes to gene flow, we also have these examples of metapopulations. These are populations of populations, hence a metapopulation. Meta, yeah. Zuckerberg ruining Facebook. Well, I don't want to get into that, right? But metapopulations is a populations of populations. So you have gene flow between these populations, which prevents the individual populations from diverging. So think about it this way. If I've got mountains like the Sandias, the Manzanos, the Magdalenas, Mount Taylor, the Sangre de Cristos, the San Mateo, I already mentioned San Mateos, I think. All of these mountain ranges would represent a metapopulation of things of different types of birds like a Steller's jay. Because you're only going to find those birds basically in the forested areas of the mountains. You're not going to find them in the surrounding desert. So we can get gene flow between these metapopulations or between the populations depending on the area of the population, the type of organism there that's moving around, the size of it, and things like that. Okay, so lakes can also be a metapopulation because they're isolated by land. But believe it or not, you can have flooding events. Rivers can flood, lakes can flow, which can connect them briefly, so you can still get gene flow. And fragmented forested areas. As we cut forest, that can also be examples of metapopulations. Now, a lot of times when we have these populations of populations, not all of the habitat is the same. Some habitats are large, some are small, and some might be degraded or might be suboptimal. So what you get is what we call a source sink dynamic. Now, a source and sinks. So if I've got a large population in a very healthy environment, it's going to start producing lots of individuals. They're going to have high success rates of surviving to reproduction. But they'll migrate out to other populations, and they might land in areas that aren't as good or are smaller. And they can subsidize those other populations that are otherwise known as a sink. So you get a source sink dynamic. Now this is important because if you go out and you start sampling, a species of birds across a landscape, and you go all the mountains and you're like, yeah, I'm finding, Steller's jays, these bright blue birds everywhere. Or I'm finding a really good one. It's called a crossbill. Their bills are crossed like that. They're pretty awesome, and you'll find lots of them. Now, interestingly, the issue comes is that if you lose the source population, then all of a sudden all the sink populations could be impacted as well. And where the bird was once abundant because you're seeing it everywhere, but if you lose the sources, the sinks will also begin to wink out. They'll go become extirpated. Sorry, and then all of a sudden you're like, wow, we had a lot of these birds and now we have none. So this is where the importance of source sink populations come in, especially in conservation. Now, when you have these source sink dynamics, high rates of gene flow will prevent local adaptation or even speciation. Lower rates of gene flow. So if I get, a species that doesn't move around as much, or think about Mount Taylor, it's 70 miles, 60 miles west of the Sandias. You're not going to get very many Steller's jays moving between there, but you'll get them. And what you'll get is maybe a little local adaptation because that mountain range is higher than the Sandias, right? So with reduced gene flow, you might get more local adaptation. You cut gene flow off, and then you can actually get speciation. So the loss of gene flow between populations, like I said, you could have local adaptations, eventually in speciation with a loss of gene flow, and also if your sink populations remain small, you can also reduce the fitness because you're not constantly bringing in genetic variability to keep it going. And then at that point, it can become extirpated, which means it's lost. So these source sink dynamics are incredibly important for these metapopulations. Okay, and sometimes we even move species around. There's a little woodpecker in the east called the Recalcata woodpecker, where they can't disperse between the populations because, well, their habitat now is way too fragmented for these populations to move around. So we'll take birds from Florida and take them up to Georgia and South Carolina, once in South Carolina and North Carolina, and all over. And that way we can maintain the health of the populations. Okay. Now here's an interesting thing. And it's hard to figure out exactly where this fits in there, but when it comes to gene flow, typically when you have individuals moving around, they're reproducing and producing offspring, which is vertical gene transfer. You can also get genes from your environment, which is horizontal gene flow. I've mentioned this before in class. 5 to 8% of your DNA is of viral origin. 5 to 8%. And that's a lot. So we're gaining genes that have clearly affected our evolution, like the evolution of placenta in mammals from a virus protein called synectin. So it's been important. It's been very important in our evolution. The other one is the evolution of eukaryotes in general, when you had the merger of the aerobically respiring bacteria into the archaean. A lot of the genes of the ancestor to the mitochondria migrated to the nuclear genome of the host, which would be another example of lateral or horizontal gene transfer. The same also happened to the chloroplasts in the evolution of plants as well. So there's this horizontal gene transfer. And in bacteria, oh my gosh, it's rampant in bacteria. It leads to all kinds of things, including antibiotic resistance. So understanding the role of horizontal gene transfer on evolution, this is current, research is being done on this, and this is part of what we need to add to the modern synthesis, because a modern synthesis doesn't take this into account, but yet horizontal gene transfer is very important. Okay, now lastly, let's talk about mutations. Mutations are any change in our sequence of DNA, and they're the ultimate source of variation. I mean, we talk about sexual reproduction, generating, generating lots of variability in a population, but it's just shuffling the deck of cards. But mutations can create new alleles, new genes, new, let me scoot here in the middle more gene families, chromosomal rearrangements, new chromosomes, and even whole genome duplication. And in fact, I was just reading this morning, a paper came out, and I should see if I can find it for you guys. But it turns out that like survivors of mass extinctions tend to have had more genome duplications than others. So, mutations, right? I mean, they're in our pop culture. Them, 1954, nuclear scientists, it's a nuclear monster film set in New Mexico where ants that were irradiated from nuclear testing became enormously large. Or there's Teenage Mutant Ninja Turtles. They're teenagers. They're mutants. They know, what is it called? Ninjutsu. And they like pizza a lot. And then, of course, the next evolution in human evolution are the X-Men with Wolverine. All right, so mutations are definitely in our pop culture there. There are different types of mutations, from point mutation to gene duplication to horizontal gene transfer, chromosome duplication, and even whole genome duplication. Okay, point mutations are actually important. A single nucleotide has been changed. A A gets switched to a T. These can be silent mutations, they can be nonsense or missense mutations. And in fact, we've often thought that a lot of the mutations that were silent don't really have any effect. And it turns out that's not the case. If you remember, the genetic code has a codon where that third position, so you've got like a TTT, that like that third T, if you switch it to an A, you still code for the same amino acid. So it's silent, it doesn't have any effect. Well, it turns out that these silent mutations might not be so silent, because anytime a cytosine gets removed or added, that can affect what's called methylation, which is a type of epigenetics. So that can actually affect how your genes are regulated. So even though you're not changing the amino acid, or you're not changing the protein or the gene, but you can change ultimately gene expression by these silent mutations, which may not be so silent, may not be so beneficial. So yeah. So at any rate, point mutations, they can be anything. Like if you look over here, you can see next to me you've got no mutation. You can have a silent mutation where you still have lysine, but you could also have TTC. Like I said, if you replace that, then that could be treated differently. A nonsense is where you have a mutation where you have lysine, you change the T, the first one from an A to a T, that gives you a stop. So what happens now is that you, instead of coding for the whole protein, you stop wherever that mutation is, because UAG is a stop codon in RNA. It tells you to stop translating. And so, for example, O blood is a mutated A that has an early stop codon in it. So you got an ATC that wasn't supposed to be there, so you don't make the A antigen in O blood. And then a missense, of course, is you replace the C with a G. Instead of having arginine, you have threonine. So these are different examples of mutations. And like I said, they can range from nothing at all to affecting how your genes are expressed to premature stop codons. And if you do that in a very crucial protein, things aren't going to go well for you. In addition to point mutations, you can also have insertions and deletions. So you can insert a letter or remove a letter, and the problem is that changes the reading frame downstream. So every amino acid downstream is going to be different. So that can cause large problems for your gene expression unless you're actually copying the entire codon, which actually does happen. You can get repeats of the codons. Okay, so we see point mutations. I gave you an example. Another one is the white and purple flowers of Mendel's pea plants. This is caused by a point mutation that basically you knock out some protein that is crucial in the making of anthocyanin, which is that purple pigment. Point mutations lead to things like cystic fibrosis, which affects oh gosh, I forgot. There's some transport protein, and you get a buildup of mucus and inflammation with the cystic fibrosis. Also, sickle cell disease and Tay-Sachs are also caused by point mutations. All right. The other one is what we call a gene duplication. So in crossing over during meiosis, specifically prophase 1, you can switch genes around. And you can get these genome or these gene duplications. This is very important because gene duplications lead to the evolution of gene families. Gene families might be blood type. Gene families might be our ability to see color, the different types of opsins for detecting light. And especially in mammals, the ability to detect odors is important because the ancestors to mammals evolved over 1,000 genes, over 1,000, to detect odors. Because every time you have a new gene to detect odor, you can detect more combinations of chemicals in your environment. And so olfaction is a result of gene duplication here. And the reason why we think that happened is because, well, the dinosaurs were the large dominant diurnal or active during the day animals. We think that mammals may have actually evolved to be more active at night when the dinosaurs were less active, so the story goes. And as a result, sight was important, but so was the ability to detect odors in your environment. So this evolution of the gene family over time allowed mammals to have like a really good sense of detecting odors. Yep, there we go. So yeah, these are the first mammals here that were detecting odors. And the first eutherian mammals were about 160 million years ago. Now, earlier I talked about horizontal gene transfer is gene flow. Is it a mutation? I don't know, because lateral gene transfer is changing the sequence of our genome. And lateral gene transfer, we get this between species, is very important for driving evolution. For example, you got these these aphids, red and green. The red ones have carotenoids in them. Animals don't have genes to make carotenoids, I think. We have to get them from our diet. So other organisms are making those from us. But the red morph aphid makes toluene, a type of carotenoid, and it does it because it got the gene from a fungus. So lateral gene transfer, but it changed the sequence of alleles and genes in this organism. So maybe it's a mutation too. We say that animals aren't photosynthetic. Well, that's not always true. One of the things that I love about biology is that there's always exceptions. Biology is complicated. And we have this thing called Elysia chlorotica, which is a type of sea slug. And Elysia chlorotica, when it eats algae, it doesn't digest all of the algae. It actually takes the chloroplast and moves the chloroplast into its cells. And I don't know how it does this, but not into its cells, but into its tissues. And it makes it green, photosynthetic. And it also has genes that have been transferred from the algae to help it maintain the chloroplast. So the chloroplasts are being maintained by the sea slug as well, which is pretty cool. This date is up for debate. The placental mammals, which are you and I, and things that are not the marsupials, we have a very long gestational period compared to marsupials. It's been thought that the first placentals may have rapidly started to diversify about 400,000 years after the extinction of dinosaurs, and that may have been due to that viral infection. However, some evidence indicates that the first placentals may date back to 100 million years. We're still trying to figure this story out, but I often say, hey, we get synectin from this virus. However, similar viral infections have occurred over 6 times in different mammalian lineages, leading to differences in the placenta. So even this story is more complicated. Okay, so I'm going to stop here, and we're going to pick up tomorrow of why understanding these evolutionary processes matter. So, man, it looks like most of the class actually jumped on at some point. Thanks for understanding. My kid woke up this morning and she was a little sick, and I didn't think it was so bad. And on the way into daycare, she's throwing up, and I'm like, no, we have to go home. Okay, so I appreciate everybody for understanding, jumping online here. This is not optimal. Come to with questions tomorrow in class, and I will answer any questions, go over anything you want me to, and we'll pick up with this understanding evolutionary processes and why they matter tomorrow. And I think we're going to have time to review for the test as well. So come with your questions for the test review. We'll get it done in class. All right. I'll see you guys later. And man, a couple of you gave me a like. Thank you so much. I'm like every other, social media influencer out there. It always makes me happy. And I will see you guys tomorrow.